Welcome everyone to the April seminar for the Cardiovascular Research Network Rising Star Seminar Series. Today we'll be having two speakers, Dr. Jessica Orchard and Dr. Michael Huang. And the theme of today's talk will be understanding our heart from cell biology to screening. So just before we go on, I'd first like to begin by acknowledging the traditional owners of the land on which we meet today. Here in Centenary Institute is the Gadigal people of the Eora Nation, and pay our respects to elders past, present, and emerging. So just a little bit about the Cardiovascular Research Network. So CVRN is formed by and there are 14 umbrella organizations in CVRN, as you can see in this institutes here. And it comprises almost 400 members from EMCRs to senior researchers. And the research that spans across biomedical, clinical, and public health. So CVRN has governance of framework, and this seminar is organized by the Rising Stars subcommittee, which we aim to assist the executive committee and also the society members to promote the opportunities for EMCRs through various opportunities, such as this seminar series. And this is the committee member, so we represent from various umbrella organizations within the CVRN. So today, the first speaker will be Dr. Jessica Orchard from Centenary Institute. So Jessica is a Heart Foundation postdoctoral fellow and then senior lecturer at the Centenary Institute and University of Sydney. She was awarded PhD in November 2020 without any emendation. It's very impressive. And also she has various she has Master of Public Health background in economics and law from Sydney University, and she became a fellow of European Society of Cardiology in 2019. So her research aims to improve the quality of cardiac screening programs to prevent sudden cardiac death, so SCD, and catastrophic disability from stroke. So Jessica will be talking to us first, and her title for today will be Cardiac Screening in Elite Australian Cricket.
So good afternoon, everyone, and thanks very much for listening today, and thanks also very much to the CVRN for inviting me. So today I'm going to share with you a few insights about our experiences rolling out a cardiac screening program in Australian cricket, together with the results of our cardiac screening audit, which we published recently. So as Peter kindly said in the introduction, my background's in public health, and then I completed my PhD at Sydney Uni in November 2020. So I'm now a postdoc, and my focus is on how to design and run a best practice ECG screening program, and part of that is improving the accuracy and the compliance. So before I get into the main part of my talk about cardiac screening of the current players, I thought I would mention a bit about what's been going on in recent weeks, because it's been a massive few weeks for cardiovascular disease in cricket, with the sudden death of Shane Warne at age 52, as well as Rod Marsh, age 74, and both in quick succession. And these tragic events have really brought cardiovascular health, and particularly cricket, into the spotlight recently. So just as we saw the Kylie effect with breast cancer, we may also see the Warne effect with heart health. So when Kylie Minogue was diagnosed with breast cancer in 2005, Professor Simon Chapman from the University of Sydney did a study which found there was a 20-fold increase in the news coverage of breast cancer following the diagnosis, and a 40% increase in mammogram bookings in the 6 weeks following her diagnosis. Simon actually noted that this could be a health promotion opportunity, and suggested that health advocates even consider developing strategies in advance for responding to news coverage of celebrity illness. So in view of this, a number of cardiovascular researchers have featured in the media lately, which I'm sure many of you would have seen. I was recently invited by Associate Professor Andre Lagersh to join him and Alex Clements in recording a podcast for the National Centre for Sports Cardiology. And I also did another couple for Body and Soul. And all of these were emphasising the prevention measures, such as heart health checks that can be taken, especially by people in those middle-aged sort of groups that really are close to the age of Warne. However, as you would know, in the younger age group, and that is for the current cricket players, the screening and prevention options are generally completely different, because what we're really looking for is screening for rare conditions associated with sudden cardiac death, rather than for coronary heart disease, which is the condition that is much more common in those aged over 35. So sudden cardiac death in sport is a rare but tragic event. However, it is the leading cause of death for people playing sport. It's important to understand that the risks vary considerably according to age, gender, ethnicity, sport, and competitive level. In Australia, the risk of sudden cardiac death for people aged 1 to 35 years is 1.3 per 100,000 per year, with 15% of these cases occurring either during or immediately after exercise. So many of the conditions associated with sudden cardiac death can be asymptomatic, and competitive sport, as I mentioned, may be a trigger. In Italy, screening of all young athletes, which is very broadly defined, is mandated by law. And the real key study that's often cited in this area showed a 90% reduction in the incidence of sudden cardiac death in the screened population compared to the unscreened population from the years before screening was introduced. However, this success has not been mirrored in Israel, which also has a mandatory program, nor in various US programs. The English Football Association, or FA, have recently published 20 years of data from screening 11,000 soccer players, and these players were all screened once per career at the age of about 16. During the period of the 20 years, there were 8 cardiac-related deaths, but 6 out of 8 of these athletes actually had normal screening results. The remaining 2 players were actually diagnosed with hypertrophic cardiomyopathy, but did continue to play against advice. This paper is really interesting, and it's been interpreted in various ways. Some people say it's evidence that screening doesn't work because of the people who had the normal results and still died, while others say it actually shows that we need to screen more often because we weren't picking them up. My view is that this study confirms the reality that even a very, very well-run program sadly will not capture all abnormalities. So while there are some differences in opinion and gaps in the data, screening of athletes, particularly at the elite level, is recommended by numerous leading bodies internationally, including in Australia, the Australasian College of Sport and Exercise Physicians. And as a result of these recommendations, many major international and Australian sports have introduced cardiac screening. So as shown in the table on the right, we conducted a survey of Australian sports cardiac screening programs, which showed that the majority did have a formal cardiac screening program. Of those sports with a program, all included a history and physical examination and a resting 12-lead ECG, but not an echocardiogram as standard. Obviously, if it was required as follow-up, that was different. About 75% of the responding sports were screening junior elite athletes as well as adult elite athletes. So it's also important to consider the underlying risk of the population to be screened, which is relevant for developing an appropriate policy. For a genuinely low-risk sport like golf, it could be argued that cardiac screening is pretty much unnecessary, whereas for a particularly high-risk sport such as basketball, a more mandatory program may be justified. So while cricket has officially been classified as low risk in this Mitchell classification here, we've actually published some data on the workload that cricketers undertake to support the argument that it's actually much more of a medium risk sport, although not as high as something like basketball or cycling. With these considerations in mind, Cricket Australia has developed and implemented a screening policy that we believe is consistent with best practice. We aim to always make it the gold standard, and we're always reviewing it to make sure it's as good as it can be. The policy was developed with our expert sports cardiology panel, and it's an opt-out model. So all new players are screened on entry into the system, and then players aged 16 to 21 are screened every 2 years, and players over 21 are screened every 5 years. People who opt to sign waivers review their decision to opt out annually. Players are then screened with a history and physical and resting 12-lead ECG. Follow-up tests are conducted if required. All ECGs are interpreted by physicians with expertise in athlete ECGs, and any ambiguous results are reviewed by our expert panel. The policy covers all male and female elite players, beginning from the under-17 state squads and extending up to the national level, including BBL and WBBL. Junior elite players included are those who are selected in national championship squads. International BBL and WBBL only players are also included, which is sometimes logistically challenging if these players arrive just shortly before they start playing, which is often during a holiday period and would make organising any follow-up, if it was required, it would make it a bit trickier. So cardiac screening results for all players are saved into the electronic central database, which is called the Athlete Management System, or AMS. This database can also generate reports for various groups and dates as required. We have made numerous changes and improvements to this database to assist with auditing and follow-up. We recently conducted and published a very detailed audit of this screening policy. The aims of this audit were to document the ECG features of elite male and female cricketers and to make comparisons between gender, age and player groups, to determine the percentage of abnormal ECGs according to the international criteria for athlete ECG interpretation, and to assess the level of compliance nationally with an elite screening program. Data were extracted from the central database for all the players covered by the audit as at January 2019. This included demographic data such as age, gender and player position. Then every player's complete cardiac file was also reviewed to obtain their screening results, ECGs, waivers, details and completeness of follow-up, and to see if there was a diagnosis or any other requirement that that player be monitored more frequently. All ECGs for players in the audit were then re-reviewed by one of 4 expert sports cardiologists according to the current gold standard international criteria. The good news is that compliance was extremely high amongst all groups. Overall, 98% of players were compliant with the policy. That is, they had either been screened or signed a waiver at the time of the audit. The mean age of the players included was 20 years, with more than 1/3 being female players. Only a very small proportion opted out of screening, and a total of 855 ECGs were available for review for the audit. In terms of conditions associated with sudden cardiac death, like the cardiomyopathies, the good news is that we didn't find any. However, it's important to note that we don't really know what the true rate would be, as we would need a larger data set over a longer time period. However, this data would suggest the rate is likely to be less than 1 in 500 in cricketers. A small number of minor diagnoses were made during the period, and they're shown here. So one of the burdens of screening is follow-up testing, and this has decreased enormously with improvements in accuracy of the international criteria. So as you can see, a small number of additional tests were required, with echocardiogram being the most common. 1% of players required monitoring with increased frequency, that is sooner than their next scheduled screening. We found a number of common, generally benign athletes' heart findings in the ECGs. Sinus bradycardia was very common and was present in about 1/3 of all of the ECGs. Left ventricular hypertrophy, according to voltage criteria, was significantly higher in males than females, and highest in junior male fast bowlers, where it was present in 28% of ECGs. Partial right bundle branch block was also most common in junior male pace bowlers. These 3 ECG features are all considered normal for athletes, I should point out. Any T-wave inversion, that is in any leads other than V1 and AVR, was present in 3.7% of all ECGs and was significantly more common in females than males. It's important to note that T-wave inversion is acceptable in some leads, but not others, and therefore only some of these ECGs with T-wave inversion were considered abnormal overall. So one of the added benefits of implementing a cardiac screening program is that it raises the profile of cardiovascular health across the organisation. There has been an increased focus on emergency action plans with a medical briefing before each game, which identifies the paramedics and the location of defibrillators and other relevant information. Also, cricket doctors now have smartphone ECGs, which can be very useful in recording transient arrhythmias, and there have been several examples where they've been really, really useful. So here's one of the examples where a smartphone ECG was particularly useful. A talented junior player had repeated episodes of tachycardia every few weeks, which were unable to be captured on a Holter. At the suggestion of his doctor, he actually bought his own smartphone ECG and did actually manage to capture a trace which showed a narrow complex tachycardia with a heart rate of about 210 beats per minute. And this was actually sufficient to lead to a diagnosis of paroxysmal supraventricular tachycardia. He's actually since undergone 3 ablations for this. So in summary, this is the first complete audit of screening ECGs in elite Australian sport and the first in the world to do this for elite cricket. Our colleagues in England have just published their cricket cardiac audit screening data, but it's nice that we definitely got in first. There was extremely high compliance with the screening policy nationally. It's a major strength that we have complete data for the entire population of elite male and female cricketers in Australia, meaning that unlike many studies, we have a denominator and are able to accurately identify the percentage screened. No players were diagnosed with a condition associated with sudden cardiac death, nor were any excluded from sport due to cardiac screening. No cardiac arrests or major cardiac incidents occurred to any players during the previous 5 years. The rate of abnormal ECGs was relatively low and very similar to that reported recently in 2 other studies. Cricketers do display ECG features consistent with athletes' heart. Several of these features, such as LVH and right bundle branch block, were more common in junior male pace bowlers, although T-wave inversion was more common in female players. These data further support our argument that cricket is more accurately classified as a sport with moderate cardiac demands. In future, data from a bigger cohort will hopefully be able to shed more light on the true rates of these rare conditions in cricketers. We're currently building a larger comparative database of screening ECGs across multiple sports, which we're hoping to build into a national registry together with the AIS. The central point overall here is really, if you are going to screen, it really must be done well. So I'd like to finish by thanking Cricket Australia, AMS staff and the members of the Cricket Australia Cardiac Experts Advisory Panel for their help, which is in particular Associate Professor Harry Raju, Associate Professor Andre Lagersh, Professor John Orchard, Associate Professor Raj Purinik, Professor Chris Samsarian, Dr. Mark Young and Associate Professor Alex Contreras. Thanks very much.
Great, thank you Jessica for a great talk. Yeah, and we've already got some questions in the chat, so I'll read them out, and if you could just answer them, that would be great. So the first question comes from Angelo. So he's asked, how useful are Apple Watches for identifying arrhythmias in young athletes?
Yeah, that's a really good question. So the newest Apple Watches also include the ability to take an ECG, a single lead, which has got an automated algorithm for atrial fibrillation. In essence, anything that can record something when it's happening to you gives you a bit more information. It might not be perfect information, but it might be a lot more to go on than what you otherwise have. And so it can, if it's a rhythm issue that you're having and it seems to come on with exercise and you have your watch with you, then that could be a really good way of recording some more information and having a much more directed conversation then with your doctor. So I think they can be very useful and are definitely part of the picture.
Next question is from Audrey. Great presentation, Jessica. Apart from ECG, have you looked at any other measurements, such as their structural or functional changes with cardiac MR?
That's a really good question. The answer is no, we haven't. And I guess one of the reasons for that is we're sort of looking at measures or screening that's already been done as a requirement for their sport, and we haven't asked people to do anything over and above what's already required by their sport. So that's certainly something you could look at, but we'd have to sort of really think of a good study question to justify putting people through that sort of extra time and investigation. Yeah.
Okay. Next we have a question from Isabella. Thank you for your interesting talk on your important work. Following on from Angela's question, do you think using smartphone ECG or wearable ECG be useful in future screening and lower cost?
Yeah, that's a really good question. It's definitely lower cost, but it's also lower information. And so I think there's a really important line between, are you doing a proper cardiac screen for your sport versus are you trying to basically use it as an event monitor to capture and document a symptom. And so I think at this point, the smart devices are very, very good at being able to capture a symptom far more accurately than just simply being able to describe it. I mean, if you can catch it on a Holter, fantastic. A lot of the time you can't. So I think that's probably where they're at the moment, because you are missing so much of the data that you would need, or it's quite cumbersome. So I believe you can actually take a 12 lead on an Apple Watch, but it takes you about 7 minutes, and you would probably need to be told exactly where to put all the watch to do that. So by the time you're up at that sort of level of difficulty, perhaps we're not quite there yet, but technology is changing really fast, and so it's quite possible we will be there at some point.
And I guess relevant question from Lisa on these devices is that, are those accredited or calibrated in any way?
An Apple Watch. So anything that's sold with that ability in Australia has to be approved by the TGA for that purpose. So, so the Apple Watch and any of the smartphone devices will have had to have been approved by the TGA, including the accuracy of their algorithm for AF. But like everything, it's not perfect. And so you can certainly see examples of it getting it wrong. For instance, if there's a lot of tremor, so if your wrist was shaking a lot, that could make it give you an incorrect answer, yeah. But they're reasonable, I would say.
We've got another question from Sally. Why did the frequency of screening differ between players under 21 compared with those over 21?
Yeah, that's a really good question, actually. And our expert panel tells us that between the ages of about 16 to 21 is when players' hearts change the most. And so there's more chance that if they did have something wrong, that it might not have been there at age 16, say, but it might have already become apparent by age 18. Whereas once they're over the age of 21, I think that any changes are much, much slower, and so it doesn't need to be as frequent.
Okay. If there are no other questions, I might just end with one question from myself. So you said there's higher risk in Black African basketball players than any other sport. So does that tell is in a way genetic driven as well? Or does genetic play a part?
That's a really good question, and there's probably some things that are not yet well understood, but the risk in male Black African American basketballers in the highest division is so much higher than in anything else. It's about 1 in 3,200. Whereas in other areas it might be 1 in 50,000. It is markedly higher, and so I think in the US they've been able to do a lot more research because it's just so much more frequent. And so I'm not sure that it's entirely well understood, but others who are more specialised in the genetics might have a more full answer on the genetic side of that, but it is fascinating.
Okay, great. Thank you very much, Jessica. I think we don't have any more questions, so we might move on to next speaker. Thank you.
Thank you.
So next speaker is Dr. Michael Huang. So Michael is a senior research fellow at the NHMRC Clinical Trial Center, and his research investigates multifaceted roles that iron and mitochondria play in pathogenesis and treatment of many human diseases. And his research spans from cardiometabolic diseases, neurodegenerative diseases to chronic diseases. And Michael's research spans from basic science to translational and preclinical research that utilizes multi-disciplinary approaches. And since 2009, he has produced 1 international patent and over 42 peer-reviewed publications with over 13,000 citations in high-impact journals. So with that, I might just introduce Michael for his talk today. All yours, Michael.
Thank you, Peter. Sorry for a mouthful there. Yeah, so thank you, Peter, for the introduction and the committee for inviting me to join this seminar. And I'd like to share with you some of my recently published finding in redox biology, looking at impaired mitochondrial homeostasis or mitochondrial life cycle in mitochondrial heart disease. So everyone here can appreciate how important the heart is as an organ, and also the amazing work that it does. We all know that the ATP is the main energy currency of the heart, and what may not be so widely known is that the human heart actually produces and consumes a staggering 6 to 30 kilograms of ATP per day just to maintain its normal function, and that's equivalent to about each beat consuming about 300 milligrams of ATP. To put that into perspective, the human body every day consumes about its weight in ATP. So despite the heart being only about 0.5% of body mass, it consumes almost 30% of ATP of the body. And the heart is actually almost exclusively fueled by ATP produced by the mitochondria. There's some flexibility in terms of the type of fuel it uses, depending on physiological or pathophysiological conditions. So it's quite adaptable in that way, and partly because of the mitochondria as well. And owing to this dependency on mitochondria, about a third of cardiomyocyte volume is actually occupied by the mitochondria. So that goes to show how integrated mitochondria is to cardiac function. The mitochondria or mitochondrion is actually a very interesting organelle. It's got its own DNA, and there's actually several copies of mitochondrial DNA within each individual mitochondrion. Some of the well-known functions include energy production, as I mentioned earlier, but also in apoptosis, in calcium signaling, and actually many other functions that govern cellular health. And owing to its role in energy production, mitochondrial energy demanding tissues, energy demanding cells, such as cardiomyocyte, neurons, skeletal muscle, beta cells, and adipocytes are very reliant on mitochondria for the energy supply. And when there's a dysfunction in mitochondria, this leads to heart failure, neurodegeneration, myopathies, as well as diabetes. A very good example of how mitochondrial dysfunction can manifest into a systemic health issue is looking at Friedreich ataxia, FA. It's the most prevalent inherited ataxia in Caucasians, and the major symptoms besides neurodegeneration is actually heart failure as a major cause of death, but also muscle weakness and diabetes. So FA is due to a mutation in frataxin, in the gene frataxin, which is a nuclein-encoded mitochondrial localized protein. Frataxin plays a role in the synthesis of iron sulfur cluster and heme within the mitochondria. And these two cofactors plays a vital role in a lot of redox active reactions. So especially within the mitochondria in terms of the respiratory chain. In terms of the cardiac pathology, because it is a major cause of death, so that's part of my major focus. It includes cardiac hypertrophy in the patients, pronounced intracellular iron deposits, extensive fibrosis, and severe decreased activity of iron sulfur cluster enzymes, particularly mitochondrial complex 1, 2, and 3. Using the heart model, the muscle creatine kinase conditional frataxin knockout mice model as a surrogate of FA patients. The model recapitulates many FA cardiac symptoms, including cardiac hypertrophy, iron deposits, and fibrosis. These mice are asymptomatic up to about 4 weeks of age, and they develop a severe heart failure by about 10 weeks of age. Using this model, my previous studies demonstrated that frataxin deficiency leads to a dysregulation of iron regulation within the cardiomyocyte, and this is characterized by a paradoxical cytosolic iron deficiency, which brings in more iron into the cardiomyocyte and shuttle directly into the mitochondria through the only known mitochondrial iron importer, mitoferrin. But within the mitochondria, iron is not being used for iron sulfur cluster and heme synthesis, and without any known transporter shuttle iron out of the mitochondria, it accumulates within the mitochondrion, leading to this accumulation amorphous iron aggregates, which is not sequestered within ferritin. And as you can appreciate, that iron is really redox active, especially within the environment of mitochondria. So without a protection within the ferritin, it can cause oxidative stress, and it does. And my subsequent study demonstrated that there is actually impaired, I guess, master defense regulator, master defense against against oxidative stress through the NRF2. That's being dysregulated within the Frataxin knockout mice. And this involves two mechanisms. One is the increased expression of its inhibitor, KEAP1, but also activation of GSK3 beta that shuttles NRF2 out of the nucleus, and thereby preventing it from activating antioxidant genes. But what we didn't know is how these dysregulation affects the mitochondrial homeostasis, or in other words, how it affects the mitochondrial life cycle. The mitochondrial life cycle is a way for mitochondria to mitigate mitochondrial stress, and it has four main processes. First is biogenesis. Then there is mitochondrial network dynamics, which includes fusion and fission processes, as well as finally mitochondrial recycling through mitophagy. So under energy deficiency, biogenesis is activated and increases mitochondrial mass. And upon mitochondrial stress, the damaged mitochondria can fuse with healthy mitochondria to decrease the mitochondrial stress through dilution and complementation. But fused mitochondria can also undergo fission, and thereby increasing mitochondrial number and compartmentalizing damaged mitochondria into daughter mitochondria, then leading to increased recycling through mitophagy. So in the FA patients, looking at the mitochondrial ultrastructure, we can see these abnormal cristae structure within the left ventricles, and there's also accumulation of mitochondrial iron within the mitochondria, just like we saw in the animal model, and also proliferation of these smaller irregular mitochondria within myocardium. Looking in the longitudinal LV sections of the knockout mice, at 4 weeks of age, we can already see some dysregulation, disorganization of mitochondria within the knockout mice at 4 weeks of age, and this include an increase in mitochondrial area, so it's now occupying more area per cardiomyocyte. It's also getting a bit larger in terms of its cross-sectional area, but it's getting shorter in terms of aspect ratio, so it's getting stubbier. And you can also see this distinctive collapse or stacking of the mitochondrial cristae. By 10 weeks of age, a lot of the aspect you see are worsened. So you can see the stacking very clearly, very pronounced in the mitochondria, and mitochondria are getting elongated, much longer compared to the wild type. And you can also see these mitochondrial iron loading. So overall, it's actually demonstrating that the mitochondrion within the knockout mice at 10 weeks of age are getting shorter but longer. Sorry, smaller, thinner but longer. It's actually there's more mitochondria and it's occupying more larger area. Looking at the transverse plane, it's pretty much the same. What's notable is at 10 weeks of age, you also see these entangle or elongation of mitochondria even in the transverse plane. So if you think of it in terms of the three-dimensionally, the mitochondria is turning into this entangled mass, and that's completely displacing the normal sarcomeric structure. So the ultrastructural study demonstrates to us that it's increased proliferation of mitochondria and potentially increased biogenesis. There's also increased fusion, leading to decreased mitochondrial area, and also increased fusion, potentially increased fusion due to increased aspect ratio. So then we'll start looking at a lot of molecular markers for these individual processes. First, looking at biogenesis. The biogenesis is mostly regulated by the PPAR gamma coactivator 1-alpha, PGC1-alpha, which regulates several transcription factors, including NRF1 and TFEM, which in turn increase the transcription of enzymes in ETC. Using qPCR, we demonstrated that there's increased mitochondrial DNA copy number and an increased expression of PGC1 alpha, NRF1, and TFAM into protein level. But in terms of activity, PGC1 alpha is actually regulated inversely by its acetylation level. And at 10 weeks of age, there's actually increased acetylation of PGC1 alpha, demonstrating that there's decreased activity. And that shows in the expression of this transcript, downstream transcript. The regulation of PGC1 alpha at the transcription level is predominantly regulated by AMPK and Paris, which are inducer and inhibitor, respectively. Looking at the protein expression, you can see AMPK is increased at 10 weeks of age, and Paris is decreased at 10 weeks of age. So together, it's causing an increase in PGC1 alpha transcription. In terms of its activity, is regulated by SIRT1, which is a NAD dependent enzyme, which deacetylates PGC1 alpha, thereby activating it. And you can see the protein expression SIRT1 is decreased, and it's corresponds to a decrease in SIRT1 activity. The staining of PGC1 alpha in the FA patient in the left ventricles, you can see the staining is increasing the FA patient compared to the control. But what's notable here is that the staining within the nucleus is actually lower compared to the control. So potentially what's happening here is PGC1 alpha expression is increasing in patients, but it's not being translocated into the nucleus due to possibly increased SIRT1 activity. Decreased SIRT1 activity. And because SIRT1 is an NAD dependent enzyme, so we then looked at, we'll question whether it is due to NAD imbalance. And not surprisingly, there is quite a marked change in NAD level. So there's a marked decrease in NAD level, but a corresponding increase in NADH levels. And this is partly due to an increase in NAD consumption. So the NAD consumption can lead to either the salvage pathway up here or the shuttling between NAD and NADH. And there's an increase in nicotinamide, which is part of the NAD consumption pathway in both at 4 and 10 weeks of age, and a decrease in nicotinamide mononucleotide at 10 weeks of age. And this is due to a decrease in the enzyme converting NAD nicotinamide to the mononucleotide, and an increase in the enzyme converting mononucleotide back to NAD. There's also a decrease in ATP expression, ATP level, and it's partly due to, it could be due to decreased complex 1 activity, which is driven by the oxidation of NADH. So when there's decreased NADH, the complex 1 activity will be decreased. Looking at fusion, as you may remember from the ultrastructural assay, you can see this elongated mitochondria within the knockout heart. The main protein of interest here are mitofusin and OPA1, which drives the fusion of outer and inner membrane, respectively. Its expression in the mitochondrial fraction, you can see that mitofusin 1 and OPA1 are increasing in the 10-week-old mice. The expression of mitofusin in the FA patients also increased, more so in cardiomyocyte adjacent to fibrotic lesions. Looking at fission markers, we have the translocation of DERP1 to mitochondria, and also MFF and FIS1. Again, you can see from the mitochondrial fraction that DERP1 is actually increased in the knockout mice. So there's increased translocation to the mitochondria, indicating increased fission, also increased FIS1 and MFF. Looking at the patient sections, there's also increased DERP1 staining and also FIS1 staining in the patients. There are two interesting molecules in terms of identifying dysfunctional mitochondria. One is the translocation of PINK1 and also the translocation of PARKin, which are recruited to damage mitochondria. Looking at the expression, the total expression is increased in both 4 and 10 weeks of age, but it's the mitochondrial fraction. You can see a marked increase in both PINK1 and PARKin in the knockout mice. And this suggests to us, because the downstream the downstream result of pink-1 pocket translocation is mitophagy. So it suggests potentially this increase in mitophagy as well. And mitophagy, several important proteins here are the mitophagy cargoes. So the proteins that gets degraded with the mitochondria. These are P62, FONC1, and LC32. In one of my previous papers, I demonstrated that LC32, P62, and FONC1 are progressively increased with age in the knockout mice. And this is due to an induction of the integrated stress response in inducing autophagy. The expression of P62 is also remarkably increased, particularly in cardiomyocyte adjacent to the lesions. Looking to assess autophagic or mitophagic flux, so there's the capacity of the cell to undergo mitophagic or autophagic degradation. We treated mice with baflomycin, which inhibits the fusion of autophagosome with lysosomes. And in mice treated with baflomycin, you can see the expression of these autophagic cargoes, which are normally degraded by the autolysosome, are increased following baflomycin treatment. So it indicates that these proteins are normally being degraded by the autophagosome, as an autolysosome. And also shows that the inclusion of mitofusin here, it demonstrates that mitochondria is part of that autophagic cargo. So in conclusion, using the Frataxin knockout mice, we show there's increased mitochondrial ion accumulation leads to a dysregulation of NRF2, but there's also a metabolic deficit in terms of NAD level and ATP. There's increased mitochondrial dysfunction, thereby activating pink-1 pocket pathways, and increase in biogenesis due to dysregulation in PGC1 alpha and increased fission and fusion through their respective proteins. And finally, an enhanced mitophagy that exceeds the capacity for autophagy to clear the cargo. So I'd like to thank my PhD student, Shannon, who does the work here, and the collaborators Natty and Sean, as well as Arnav for sending the patient samples and the funding agencies. So thank you for your time.
Great, thank you, Michael. Yeah, that was a great talk and very interesting. While people maybe put up the questions, I'll start off with some questions. So the phenotype you see between 4 weeks and 10 weeks in your Frataxin knockout mice, it's quite dramatic, considering that at 4 weeks is when the disease starts to onset.
Yep.
Do you think that like a sudden, I guess, progression of disease is just really due to the age, or some sort of like stimuli, given that 4 weeks in mice biology, there's a lot of changes that are going on around that age. So like some stuff I can think of is, they'll start going through the puberty around that age, and they are weaned around that age. So I wonder these stimuli are promoting the disease. Do you have any idea on that?
Well, we don't see any difference between the male and female mice. So, so the gender is probably not an issue, and so thereby puberty may not be a main factor. And the other main thing is to know that 10 weeks of age is actually relatively young for mice as well. Mice grow to 1 or 2, almost 2 years of age, I think. So 10 weeks of age is really, really young. These mice are still relatively young mice. So it is quite a rapid model, owing to a complete knockout of frataxin rather than in the humans where there's decreased expression. So basically, bottom line is, I think a lot of these dramatic changes between 4 and 10 weeks of age is due to the deficiency of frataxin and the role that it plays in mitochondria, and how the importance of mitochondria to the cardiac function. So that's, if that answers your question.
Yeah, okay. But then if you leave it, are these mice viable after 10 weeks? Like, how long do they live?
No, they die at 10 weeks.
They die at 10 weeks, okay.
Pretty much very, very well timed.
Yeah. Okay.
And by 10 weeks of age, we did echo on these mice. By 10 weeks of age, the heart's barely pumping at all. The fractional shortening, I think, is about like 6%. So.
Okay. And we've got a question from Isabella. Thank you, Michael, for this interesting talk. What is the prevalence of FA in Australia? The Frataxin knockout mice appeared much smaller than wild type, but they have much larger hearts. Is this the case in FA patients as well?
The prevalence overall worldwide is about one in 30,000, 50,000 around there. In Australia, I think this network is about close to 1,000 patients in Australia. And in terms of mice, and Isabella, you're right that the mice are the knockout mice. The picture I showed were of 10-week-old mice. So the mice are a lot smaller because they are fatigued and diseased, but their heart is massive. The heart is, as you can see from the histology slide a few slides ago, the heart weight to body weight is out of scale compared to the wild type mice.
Okay. Maybe I can ask another question, just this more technical question about your mouse model. So you said this is MCK conditional. So what cell types are these knocked out in?
Yep. So because MCK muscle-creating kinase, you can also see that in skeletal muscle as well. So Frataxin is also knocked out in the skeletal muscle, but we actually don't see any pathology in the skeletal muscle, or at least we're not able to see that within that 10-week time frame.
Okay. Yep. Okay. Yeah, great. If we don't have any more questions, we might just thank both speakers, Jessica and Michael, again for interesting talk. And just before we leave, I might just share a screen for next month's seminar. Sorry, Michael, would you mind stop sharing your screen?
Sure.
Thank you.
All right. Thank you, Peter.
Yeah, so yeah, thank you everyone for coming to this today's seminar. So as you know, this will be the monthly seminar, and for the month of May, we'll have another two EMCR speakers, Dr. Julie McDonald from University of Newcastle and Dr. Kavitha Mithya from Victor Cheng. And this seminar will be convened by Dr. Wembo Peng from UTS, and please look out for the email for the invitation, and looking forward to see everyone there. Thank you, everyone.